Hello, hi. I'm Surya. I'm from the Simons Foundation. I'm at the Center for Computational Biology. Today I'm going to talk about one of our recent works on studying microtubule organization in human spindles. And I want to first give a shout out to our collaborators at Harvard. That's Colum. So most of the light microscopy data I'm going to show today is actually from him. And Dan for actually being the lead of this project. So firstly, what's a spindle? A spindle is a subcellular structure that basically segregates chromosomes during cell division, right? So it's a subcellular apparatus. If it doesn't function properly, it can lead to aneuploidy, which in fact can lead to cancer or birth defects and whatnot. And most importantly, if you look at different stages of cell division, they can all be characterized by the form and shape of the spindle. For instance, at the pro-metaphase, you have the spindle that's just being formed, and at the metaphase is fully assembled and is in the state of dynamic steady state. And at the anaphase, it has segregated the chromosomes and it's starting to dissolve into the cytoplasm, right? Today I'm going to focus on the metaphase stage, where the spindle is fully formed and is in this dynamic steady state, and I'll let you know what I mean by that. And depending on the organism, the metaphase can last for from few hours to few minutes. In this human spindles, for the HeLa cell lines, the metaphase lasts for 5 minutes. So most of our analysis will be focused on data that has been acquired during this stage. Okay. So if you look a bit closely at the spindle, what is it composed of? It's composed of microtubules and molecular motors. Actually, in HeLa cells, you have close to 10,000 microtubules and even more number of molecular motors. And again, apart from the molecular motors, you also have these passive cross-linkers, all these kinetochore proteins, and the chromosomes, all embedded in this rich matrix, right? And also, one of the intricate aspects of modeling such a system is that you have this huge diversity of timescales, right? For instance, you have binding events, motor binding events, which are an order of a few milliseconds, and you have microtubule turnover, which is of a few seconds, and metaphase is in a few minutes, right? So you have this cascade of timescales, and it's also very difficult to acquire measurements on the spindles for many problems. But a good modeling exercise is missing here because we have just no data on the systems. Okay, so this has changed recently because thanks to our collaborators in Germany, so they were able to do a full 3D reconstruction of the HeLa spindles at the metaphase stage. And what you see here is all the microtubules. I'm just showing you, I've rendered only close to 20% of the microtubules here. And I've also labeled two kind of microtubules. The one that are in red are called the kinetochore microtubules, which are basically the ones that are actually segregating the chromosomes. And you have this, so to speak, the other fabric, which is the spindle microtubules that are marked in blue. And the proportions, kinetochore microtubules are 15% of the total microtubules, and the spindle is mostly formed by all these blue microtubules. And for the case of this presentation, we don't really distinguish between these two microtubules, and we just model the microtubules as a whole, right? So what are the questions we are kind of interested in? The first question is can we use this data to actually probe the physical basis of the spindle, to ask the question of what are the dominant physical mechanisms that are basically driving the microtubule organization in the spindle? And secondly, can we use a class of active matter models that Margaret alluded to in the early talk and actually measure some material properties of the spindle based on this model, right? So let's just jump right in. So firstly, from this data, what can be quantified? It's very obvious, there are two things that kind of pop out, right? So we know the exact trajectory or the contour of the microtubules, and we know the position of all the microtubules in the spindle, which means we have a direct measure of the density of the microtubules and the conformation, which is basically the orientation of the microtubules with respect to some axis, right? So we know the density of microtubules in space, but not in time because it's static, so there's no time. And the orientation of the microtubules, which is basically the angle a microtubule is actually forming with respect to, let's say, the spindle axis, which is the horizontal axis here. On average, this is something you can just vaguely say about these two state variables, that the density is almost constant to a linear approximation, and most of the microtubules are aligned along the spindle axis, meaning that if you were to look in the middle of the spindle, they're all horizontal. Okay, so this is kind of me quantifying how aligned they are. So 1 means they're all horizontal, and any value close to 1 is basically they're roughly horizontal. Okay, and so you can actually take a step further and actually project the orientation of all the microtubules onto a 2D plane, and this is the representation of it. And as you see, all the microtubules in the middle are almost horizontal and nicely aligned with respect to each other. And as you see here, you almost have like a defect points here, and here you have like 45 degrees alignment of the microtubules and whatnot. And this is basically the same data, but averaged over 3 such reconstructions, not just 1 reconstruction. So you have, so to speak, some statistics to average over, right? So firstly, we can ask this very simple question, for this very simple orientation field, what is the mechanism that's basically governing this orientation field? And again, I'm not going to get into the details of things, but if you take the active matter models or actinematic models and in all its glory and actually ignore the hydrodynamics and also assume that the microtubules are highly aligned, you can actually reduce all this complex active matter model to this very simple equation, which is basically the Laplacian of the orientation at steady state is equal to 0. Okay? So all you need to do is solve this equation with certain boundary conditions, and you're able to recover almost very quantitatively the average orientation of the microtubules in the spindle, right? That's good, but for instance, EM actually, the electron microscopic reconstructions actually give us measurements at individual microtubule scale, which means we also know how an individual microtubule actually deviates from the average, right? So now the question is, can we push the theory to also explain the deviation from the average? So that's what we did. So we know the average, which is basically, so to speak, the Laplace delta n is equal to 0. That's the equation that we assume it to be the average. And we also know the individual microtubules and how they are deviating from the average. And we can actually push our active matter model to actually also explain, so to speak, all the deviations. And this is the equation. And to give an intuition about this, this basically says that microtubules are locally aligning each other, meaning that if there is a deviation of a microtubule, it only affects the microtubule next to it, and the effect actually kind of decreases as you go away from that microtubule, right? So the effect kind of diminishes as a function of distance. And in Fourier space, all the mumbo jumbo, but the thing is, you can actually derive this very nice correlations, which is basically, as a function of time, it actually decays as 1 over omega square in Fourier space. And as a function of space, it actually decays as 1 over q square, which is basically the wavelength that's being set by the medium. Okay? And this is the prediction from the theory. We can actually see if this is true. So again, what I do is I go and solve this equation in this geometry, and I take the data, which is basically the EM data, and I basically subtract it, so I get a deviation, right? And from this deviation, I basically compute these correlation functions, which the theory predicts. And what you see is this nice q square scaling that also emerges from the data, meaning that whatever the model that we assumed, which is purely based on local interactions, is sufficient to explain, so to speak, the orientation deviation data in the spindles. Okay, this is good, but the question is, EM electron microscopy is full of artifacts, right? So how much can we trust the data? So for this, we also did some pole scope measurements, basically light microscopy measurements, which actually also can also quantify the orientation of the microtubules in the spindles, but not at the resolution of individual microtubule, but a bit more coarser, right? And the good thing about light microscopy is that we can also get temporal correlations, because we also measure dynamics over time, which with the EM you cannot, because they are static. And from that, we can also make a prediction for how the correlations in time also should decay, and they very well match with the experiments. And if you now overlay the pole scope or the light microscopic data with the electron microscopic data, you see that there's a nice agreement between those two different modalities. So this gives us a lot of confidence, that we are not measuring some artifacts, but actually measuring the fundamental principles that's in the system. Okay. So in summary, microtubules, you can actually say that they align each other locally, and the exact mechanism through which they align each other could be through static interactions or through molecular motors or at passive cross-linkers. Okay. So now we can also look at the other field, which is basically the density of microtubules, right? And you can write a very simple equation for how the density of the microtubules will evolve. This equation has 3 fundamental contributions. That is, microtubules diffuse. They move along the streamlines that are actually set by the previous field, which is n, and microtubules grow and shrink, right? That's what the equation says. And again, you can do all the mathematics, and you'll actually come up with relations that actually predict how the correlation should decay in space, okay? So I'll break it down for you. This is basically, there are three competing mechanisms: diffusion, pneumatic alignment, and nucleation, right? And if all these three principles are dominating, you should see a decay that actually looks like this. And if the nucleation is the dominating force, you should see a correlation that's almost a plateau. And if you see the other way around, you'll see a 1 over q power of 4. And if it's somewhere in between, you see 1 over q squared, right? So we can actually see where the data actually sits, which actually tells us which physical phenomenon is important or not, right? And what we actually see in the data is actually this very mixed signal, which basically says that all the three physical phenomena are kind of important: the nucleation, the diffusion, and also pneumatic alignment, right? And we are able to fit to the model, and also the model has parameters, which are real interpretable parameters, and we can also go and infer these parameters just from this data, right? So now we have this model, now, so we said what can we do with this? Can we do a prediction that is completely, once we infer the parameters, can we fix the parameters and just make a prediction, right? For this, we basically asked, so what if, can you explain, so to speak, the density inhomogeneities in a plane in the spindle? And again, we can do mumbo jumbo there. And what you actually see is that without any parameter tuning, you can actually explain all the density inhomogeneities along a cross-section of a spindle, right? So this also gives us a validation for the theory that it is sufficient to explain both the density and orientation fluctuations in the spindle. Okay, so in summary, so we have just shown using a very simple theory that microtubules locally align each other, and that is sufficient to explain all the orientation field averages and also the deviation that's actually present in the data. And secondly, this cause for alignment can be many things, right? It could be steric, it could be cross-linking, or that is something that we should probably measure through other different means. But for now, we are not making any conclusions on what is actually causing the alignment. And we are also able to predict quantitatively how the densities change across a cross-section of the spindle without any parameter tuning, which also gives us a lot of confidence on our model for this concentration, which is basically microtubules diffuse, microtubules move along streamlines, and microtubules grow and shrink at a certain rate. And they're all important to explain the concentration data. And what I haven't shown today is that since we have the microtubule trajectory data, we can also measure the bending fluctuations of individual microtubules and actually measure the activity in the system, that's actually caused by all these active motors, which I haven't presented today, but we are trying to push in that direction. This combined with some microrheology experiments, we are trying to quantify the viscoelastic properties of the spindle too. With that, I would like to thank you, and here's just a plug for the Flatiron Institute. We have some postdoc positions open at all scales, so if you're interested, please do apply. Thank you.
Thank you so much. We have time for maybe one question.
Hi.
Yeah. So the whole fluctuation stuff you showed us was in the blue part, or is that true as you slide the window throughout?
So it's a coarse-grained model, so it's true in the middle of the spindles, in the spindle ball.
Okay. And so,
yeah.
So do you have a good intuition for what sets all the different directions? Because I was kind of surprised about how sharp the transition between the colors is.
You mean?
Well, yeah, the
Sure.
ones that are. No. When you do the mapping of the relative orientations, like the ones 45 degrees angle.
Yes,
degrees.
I see.
Yeah,
what you mean.
that
This
one,
yeah.
exactly.
Yes. Actually, yes. So what I haven't spoken today about is actually the shape of the spindle, right? And the shape of the spindle is actually set by the surface tension and the bulk, so to speak, pneumatic elasticity that's trying to push out things, right? So it's the competition between these two that actually sets the interface. And that's where you actually see this microtubules kind of bending along. And yeah, it's very sharp, I agree, yeah.
Thank you so much.
Thank you.